My name is Diana Gomez. I will start the last day of talk of the annual International Conference to Brain-Inspired Cognitive Architecture for Artificial Intelligence. I will be the chairman during this presentation block. We shall start with Anastasia Piven's presentation. Anastasia, are you ready to share your screen?
Hello, I'm going to be sharing the screen, Ksenia. I'm with Anastasia. We've talked about, we sent an email that I'm going to be the presenter. But I'm ready to share the screen. Can I?
¿No está Anastasia?
Sí, pues,
¿No se escucha? A mí se me quiere la chica. No sé qué soy el latino. Ya estás todo.
okay. Anastasia, can you share your screen, please?
Yes.
You have 15 minutes to present your work, followed by 5 minutes of the question from the audience. Anastasia, you can start, please.
Yes. Hello, everyone. My name is Ksenia. I'm going to be presenting the research that we've made with Anastasia Piven and Professor Jan Troel on the base of Free University in Amsterdam. And the topic of our research is an adaptive network model for aggregation and Prion-Like propagation of α-synuclein in Parkinson's disease. So Parkinson's disease is considered to be synucleinopathy. It's one of the main neurodegenerative diseases, which is defined by the loss of dopaminergic producing neurons in the region of brain called substantia nigra. So the Parkinson's disease is unspecifically characterized by symptoms such as bradykinesia, rigidity, and rested tremor. And the protein called alpha-synuclein is thought to play the key role in the development of this disease. So we have protein monomers of alpha-synuclein, which are normally present in brains, but in some cases they gain an abnormal formation and leading themselves as prions, which means that one specific monomer or oligomer might cause the other proteins, normal proteins present in brain, to gain an abnormal conformation, what leads to formation of Lewy bodies and therefore the loss of neurons and brain damage. But along with alpha-synuclein, growing evidence exists that lipids with alpha-synuclein play a role in the development of neurodegeneration. And what's going on is lipids and alpha-synuclein, they aggregate together, wasn't just simple aggregates or clusters of alpha-synuclein and lipid vesicles. It leads to alpha-synuclein/lipid dyshomeostasis. And a few mutations in specific genes were found to possibly play a role in the development of this abnormal formation of lipids and alpha-synuclein. And so after the aggregation of alpha-synuclein with lipids exists, the 4 postulated ways of further propagation of alpha-synuclein in brain might take place. So the first one might be through synapses or synapse-like structures, through nanotubes, which is basically bonds, so connection between one cell and another, incorporation by simple endocytosis and exocytosis, so vesicle inside or attached to alpha-synuclein, leaves one cell and goes into the another, and also through receptor-mediated uptake. And so based on the background information, we've created the conceptual model for our network. So we start off with the mutation in enzymes that are responsible for lipids present and mutation in alpha-synuclein responsible genes present. So the first box here, which is for enzymes, leads to enzyme dysregulation and leads to lipid dyshomeostasis. And the same holds for alpha-synuclein, which leads to alpha-synuclein homeostasis. And at this stage, different risk factors might intervene or make it worse. And so this homeostasis of alpha-synuclein and lipids leads to alpha-synuclein gaining toxicity, what leads to further seed formation, meaning this seeds of alpha-synuclein with lipids might leave the cell and through the propagation ways I've talked about, and the aggregation itself would also lead to seed formation. And so at this point, different protective factors might possibly intervene, which we don't know exactly how they do this and what exact protective factors might exist. A bunch of studies have shown that some interesting protective factors might exist. For example, smoking or coffee consumption or ibuprofen consumption might play a role as a protective factor in neurodegeneration development. And some risk factors such as infections or dairy consumption, as well as metabolomic syndrome, and as well as different medical conditions. So after the seeds were formed, four our postulated ways of propagation take place: synapses, receptor-mediated uptake, propagation through nanotubes, and exocytosis, endocytosis. And together, they are collecting to the alpha-synuclein general level of spreading in the brain, which leads to neurodegeneration. So now you can see the 3D model of for our simulation, and on the base level you can see the conceptual model, and you can see also two higher levels. Second adaptive level has 4 V states. Let's see what they mean. For example, lipid homeostasis leads to alpha-synuclein toxicity, but both the more lipid homeostasis dysfunction we have, the more alpha-synuclein toxicity we have, and they both gonna result in more alpha-synuclein toxicity. The same holds for alpha homeostasis dysfunction and alpha-synuclein gaining toxicity. The more alpha-synuclein dysregulation we have, the more alpha-synuclein toxicity we have, but they both going to result in more alpha-synuclein toxicity itself, and it holds for every V state we have. For example, aggregation and seed formation, more seed formation as a result, and of course, the general level of alpha-synuclein spreading in brain and neurodegeneration. And on the third level, you can see 4 V states which stand for speed factors for each of the V states we have respectively. So now going to the cases, to the simulation itself. So we've created 2 simulations for 2 different cases. And so the first scenario is when we have mutation in genes of alpha-synuclein and enzymes responsible for lipids. And so these mutations are present. And we have as well protection factors, but they are not sufficient to do anything with the start. So with the start of neurodegeneration. So you can see the simulation here. One by one, these lines going up represent the start of neurodegenerative process. 4 horizontal lines over here stand for different ways of propagation of alpha-synuclein, because the recent studies have shown that the different methods of spreading of alpha-synuclein in brain, they have different levels and they are responsible for different fractions of general level of alpha-synuclein. For example, the one that is thought to be the least important is simple exocytosis, endocytosis line over here, and the most important one is thought to be receptor mediated, which you can see up top over there. And so the level of aggregation stays the same after it has been put to the maximum it can be in specific cells. And the repetitive lines over here, these are graphs created with step modes to represent risk factors and protection factors. Let's look closer to this simulation here. What we can see is when risk factors drop, the level of lipid homeostasis disorder and alpha-synuclein homeostasis disorder also drops just a bit. what means that when risk factors are not present at the same high level and neurodegeneration is already present in brain, lipid homeostasis and alpha-synuclein homeostasis might be slightly better than it was before with risk factors, but it's not sufficient for neurodegeneration with a which is an orange line over here to change just a bit in any side. And protection factors, which is a blue line over here, they might overlap with risk factors. And you can see that when protective factors drop, they are not present. The level of alpha-synuclein homeostasis and lipid homeostasis disorder slightly goes up. And you can see that by the absolute values of this growth or drop, you can see that the drop when risk factors are not present, it's more in absolute value than the growth in protection factors drop, meaning that risk factors play a more significant role and they intervene in higher levels than protection factors when we have neurodegeneration present in brain. And of course, when these both things are present, we cannot reverse neurodegeneration. That's why it continuously stays at the same level. Now moving on to the second case, the scenario is that mutation for alpha-synuclein genes and enzyme genes are still present, but protection factors are predicting an older age of onset. And you can see the simulation here. So the lines for the spreading, they are the same, but neurodegeneration, the last orange line we had, is growing slower. It means that neurodegeneration is still existing in brain, but it predicts an older age of onset. But it's hard to tell how much older, because the patients with gaining Parkinson's disease, they are already old. So the difference between either it was because of protection factors or because of risk factors going up or down is hard to tell between this one or either the previous case, but we had less protection factors, or it was just a case with a person that it was already too bad for them or too good. So it's hard to tell either this neurodegeneration going slowly of how much of older onset we can talk about. And so in conclusion, we have a model now that unifies the processes of aggregation and further possible propagation pathways of alpha-synuclein. And the main focus was on alpha-synuclein and lipids, which is a hot topic right now in neurodegeneration and Parkinson's disease, and how homeostasis of alpha-synuclein and lipids can be alterated by different risk or protective factors. And the received patterns present different ways of how risk and protective factors can play roles in neurodegeneration, although we still don't know the exact patterns, and that could be a topic for future research. Thank you very much for attention.
Anastasia, thank you so much. Now we proceed with the question from the audience. If any has any question, please raise your hand. There doesn't seem to be any hand raised. So thank you so much, Anastasia, for your presentation.
Thank you.